So welcome everyone to the North Central IPM Center Pests and Progress series. My name is Laura Isles. I'm one of the co-directors of the North Central IPM Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection Pest Management Program. We have a mission to coordinate IPM across the North Central region by sharing and promoting IPM research, extension, and teaching activities. If during today's webinar you have any questions, please type them in the Q&A part at the bottom of your Zoom screen, and we'll go through all of those questions after the presentation. Today I'm excited to have Ashley St. Clair from Corteva Agriscience. She's going to talk about AI-driven image analysis for increased accuracy and precision of honeybee colony health and productivity and regulatory field studies. So welcome, Ashley, and I will let you begin your webinar.
Thank you so much. After working so often with the North Central IPM Center during my graduate career, it is such an honor to be invited back to give a webinar. So just sincerely, thank you for having me. Ashley St. Clair, like Laura said, I'm with Corteva Agrisciences now, been here about 3 years as a regulatory ecotoxicologist. So I oversee a lot of the environmental safety studies for any of our new product registrations, and also will build the regulatory dossiers associated with those submissions. And that would include a lot of our lower tier laboratory studies, but in many cases, always for any insecticidal product and can be triggered for other types of product registrations. We will conduct a higher tier pollinator field study. So that can be a tunnel study with honeybees or up to a full field study where we are able to assess what any potential adverse effects might look like under expected environmental concentrations of a product. And so in these studies, the endpoint that we are assessing typically is a measurement of honeybee colony strength. So that colony strength is going to be the numbers of adult bees and developing brood, so eggs, larvae, and pupae that are covering each frame and then calculating that out at the population level. So this is to meet a specific protection goal that's set by the European Food Safety Authority, and I would say they're the most conservative goal is that risk managers basically agreed that the maximum level of colony size reduction in a treatment scenario compared to our control colonies should not exceed 10%. However, this being said, there's not really a very clear or strict way in which these assessments of colony strength are currently required to be conducted. So the OECD guidance, so this is the Organization for Economic and Cooperation and Development. So this is a global policy forum, includes about 100 countries in Europe, says in their guidance documents, the strength of the colony should be estimated through percent comb area covered with adult bees, and that essentially we need to be doing these estimations in a recorded and quantified manner. So EPA has some similar language in their guidance document for exposure and effects testing for bees, but they have a little extra line in here where they say, where possible, the study design should rely on digital image analysis. But this isn't always possible because there are not always analysis systems in place for this. So there is some precedent already in place in some cases. So our brood development studies that are done, there are some really great papers that have come out and developed an image analysis system that it specifically allows you to track brood development. So you'll get a frame, you'll mark each individual cell, and then you're able to track the development of that cell over time. But this type of system doesn't currently exist at the colony level for population level metrics and multiple metrics within a colony. But we have precedent where regulators are currently accepting image analysis systems. So this really begs the question about how are we currently assessing this measure of colony strength or adult bee population in our studies? So to really answer this, I wanted to go over first a little background information about just highlighting a few foundational papers that kind of led us to the point that we're at right now. So in the mid to late 80s, some really great work was done where an observer estimation method was developed. So essentially starting with 2 hives hives, and they tracked about 9 frames throughout the season. In this study, they were able to develop a method where someone could just visually observe a frame and say, okay, here's the percent coverage on that frame, and convert that to a number of bees. However, some of the findings from this study essentially said that the estimate of the number of bees by the observer was often too low. So for a little background, the way that these estimates were done is that they had about 250 bees that were weighed to get an average bee weight. And then from that average bee weight, they would take a frame that's like maximum covered in bees, brush those bees off into a bucket of some sort, and then weigh all of those bees so they could get a potential upper threshold of a maximum number we would expect to see on a full frame, and then basically back calculate to figure out, okay, if it's 50% full, how many bees would that be? But we're finding that up to 16% lower than the actual weighed number of bees, their estimates were underrepresenting. So this ultimately led to having to apply a Swiss fudge factor. This was primarily because they were using Swiss comb, which is a little bit different size than a standard comb, but ultimately resulted in when the beekeeper made an observation of an estimate coverage, they had to add 100 bees to the estimate at the end just to get back up to that standard number that they were expecting to see based on the weight. And they also reported that in specific instances where bees were feeding within the cells, so if you were to smoke a colony beforehand, many of those bees are going to stick their heads in the cells to try to eat some honey as a defense response. And they were finding up to a 300% error rate where it's not necessarily a quick and easy observation if all of the bees are in cells to tell how much coverage is on the frame. But ultimately, this method was really great when it came out because it allowed really quick access to the bees, really quick determinations without major disturbance, and was relatively accurate compared to anything that we had available at that time. So that method is now very commonly used, referred to as the Liebefeld or Liebefelder method, where a beekeeper is going to pull a frame out, basically make a quick assessment. Oh, this looks like around 60% coverage, and then back calculate to solve for X, where the denominator in this equation here is going to be that upper threshold maximum number of bees that we would expect to see on a frame based on that weight. And we now have some really great information that has been collated in the Colas Bee books for standardized beekeeping practices that allows us to change this denominator number based on your region and what type of frame might be common in that region, and the maximum number of bees that you would expect to see so that you can get a little bit more accuracy depending on where you're located and what type of equipment you're using. However, all of this being said, I think these are really great methods. There's just a lot of evidence that goes to show that humans are really bad at estimating percent coverage of pretty much anything. This has been shown very heavily in human health studies like pathology reports where they're estimating coverage of cells or damage in some way. This has also been shown in agricultural work where we see percent leaf damage from a crop pest, for example, is really difficult for the human to accurately assess. And this is generally related to the fact that we as humans have unconscious and conscious biases. So some examples in the case of estimating bee coverage might be that we would expect an effect perhaps when reading a treatment frame because we know it's a treatment, or expect our controls to look stronger because we know that they're supposed to be our quote unquote healthy bees. So we can try to nip this at the bud a bit by keeping treatments blinded, et cetera, but there are always going to be certain biases that go into this type of estimation method, as well as weaknesses in just having human assessors. So things like being more attentive earlier in the day and then getting tired and kind of quick speeding through things as you work through the day, or you have allergies or some kind of life event that has you a little more distracted than a standard day. And especially when it comes to having different observers, everybody is going to have their own threshold for level of training and their reading biases. And so having a system in place that kind of can bypass all of these is essential. And then the method itself, the observer method, has some inherent flaws where up to 300% variation if bees are feeding can be a problem, especially if you're going to smoke your colonies before going into them. The fact that the method itself required a calibration fudge factor in order to get accurate results, and also that it was never truly calibrated against an actual count of the bees, but rather a average weight of bees. So I wanted to take a minute to put you all to the test a little bit. Unfortunately, I think that the chat system is shut down for just open chatting. I was going to have you just drop your guesses in the chat, but just take a second individually to look at this frame and see how well you think you can estimate percent coverage on a frame. So some of you may have some beekeeping experience, maybe some of you not. That's quite okay. Just take your guess at what the percent coverage on the frame is, and then I will reveal an answer to you in just a moment. So for this frame, we had 137 bees, so this is an 11% coverage, but this can be challenging when the bees are individually spread out across the entire frame for us to mentally cluster them into a specific amount of area. So similar here, give it a try for this frame. What do you think the percent coverage is? Okay, so based on the Liebefelder equation for a standard Langstroth frame, this would equate to about 51% coverage or 619 bees. And I have one last one for you. So for this frame being almost fully covered, but definitely still having some open spaces, one of the things I like to note is that when the frames are totally covered like this, we often get a layering of multiple bees on top of each other, which can make it challenging to estimate. We had a 99% coverage based on the equation with a count of 1213, whereas that maximum was 1,215 bees. And so I'm willing to bet that across all of you, there was some variation in your guesses. Every time I have done this, I've seen usually about a 20 to 30% swing in guesses. So this kind of demonstrates that variation in individual observers. And if you have multiple observers on a study, this could lead to some variation in the overall effects in the colony, which could lead to misleading or misidentified potential effects. So our objectives were really to enhance the accuracy and reliability of honeybee population counting and colony health measurements through an AI machine learning image analysis system that would be more GLP compliant. And we wanted to do this in two ways. One for just counting the adult bee population, and then one where we can remove the adult bees from frames and do a colony organization, so counting capped brood, uncapped brood, nectar, honey, etc. So in order to develop these systems, we collected around 650 high quality images in the field. These were split into training, validation, and calibration sets, and ultimately annotated around 400 photos. So for our bee counting systems here, we annotated individual bees on the frame. And then for hive counting, we would go through and have a skilled annotator identify pollen, uncapped larvae, capped larvae in every single cell on the frame. And so essentially the way that these systems work, and I am no expert in the true development of the system, this work was done with myself and a huge team of people at Corteva. But just to give a high level understanding, because the AI systems can tend to be a little bit of a black box. For our bee counting system, since we're only counting one measure, adult bees, on the frames, the images are basically our ground truth annotation, like the photo I just showed, is fed into the model. It's broken down into several layers called convolutional layers, where that complex data is broken up into sections. And then that is attached to a decoder section where the models, we've said, okay, this, when you see this, it equals this, and it recodes it back to the image and then gives you an output that tells you the exact location of every bee. We have a similar system for our hive counting model, but it's a little bit more complex since we're asking this system to identify multiple features on a frame instead of just one. We have to feed in kind of a mask here that is a map color-coded answer key for each pixel type. So this is commonly done in mapping systems. So you can see kind of a satellite image map here goes in. It's encoded the same way as our convolutional layer, and then it's decoded at the end, and it gives you, based on the map we feed it, a prediction result that is color coded. And this is done for each pixel in the frame, and then it recreates that picture at the end. So to get into some of our results in the models, these are still being developed right now, but we're seeing a really positive results so far. For our bee counting system, which is a little bit more developed, when we compare our ground truth annotations here on the x-axis to our model output on the y-axis, we see a really tight relationship, often getting greater than 95% accuracy, a 98% correlation here in those outputs, so really strong relationship. But you will notice that that relationship does start to loosen up a little when there are more than 1,000 bees on the frame. And I'll get back to this here in just a minute. But we also wanted to compare this model output to the previous method that was being used. So we asked a beekeeper to basically take our images and do the same assessment they would be doing in the field in our studies, and compare what those results look like. And when we look at the beekeeper counts, you can definitely start to see some really interesting trends come out. What I notice most here is that if you look at certain areas, there's a line, a clumped line of estimates here, here. So what is generally happening here is just another example of that weakness bias system where we as humans like even numbers. So we tend to just lump things into a 10% or 5% variation. Instead of looking at a frame and saying, yes, I think that's 97% coverage, we would say 90% or 95 most likely. So we tend to see these clumpings in our beekeeper estimates that lead to a greater amount of variation in overall estimates. So when we actually go through and compare this, we were consistently seeing that the beekeeper count was significantly lower than both the ground truth, where we manually counted every single bee on the frame, and the model output predictions, whereas our model output and ground truth were not varying from each other. And if we hone in on this area where I said, okay, we have a little more variation in our system right now when there are greater than 1,000 bees present on the frame, and compare that coefficient of variation for the model output to our beekeeper estimates, we see on average around a 3% variation, and up to 14% when our standards for the systems are that our percent CV needs to be less than 15%. And this is a similar standard that's used for many types of studies. Whereas our beekeeper estimate here, we're seeing on average around a 30% variation. So if we're only able to, if our threshold for the specific protection goal, excuse me, is 10%, and we're adding in 30% variation in estimates, this could definitely lead to some false errors in here. And we were actually seeing up to a maximum percent error of almost 300%. So similar to what they were seeing in the original foundational papers for this work. So one thing that I wanted to note is that when we look at the output coming from our models, it's not a foolproof system. But we are seeing a pretty good accuracy in the numbers of bees feeding in the cells. So you can see instances in these circles here where bees are down in the cells. This is a scenario that was more challenging in the observer method, and we are able to capture those bees pretty reliably. But like I said, it's not perfect. So here's a couple examples of just a few places on the frame where the system failed to capture a couple of bees. But overall, seeing that variation be quite low compared to our ground truths and to our beekeeper estimates. Um, Moving a little to our hive counting model now. So this model is still under development, but can give you a little bit of feedback on where we're at with it right now. So we have gone through and annotated many of our images. And when we first started these annotations, we would often see large areas of a frame. So up here in the upper left-hand corner, these are mostly all empty cells. And so these would be masked as one giant mask of, oh, these are all empty. These are all white open brood here, capped brood, etc. And then we were asking the model basically to make those predictions, and we were finding that those predictions were not coming out clean. So there's some little chopped areas here, some confusion in the model, and we really were trying to troubleshoot what was going on here because we're finding that any instances where the cells are open, uncapped nectar, uncapped larvae, pollen that's not capped, we're seeing 78% accuracy, 67, 73. So we're not in this 90s range that we would like to see, even though our overall model was performing pretty well. And so this really begged the question of what exactly is going on. And this really highlights that point that quality information going into a model is what is going to result in your quality information coming back out. So each trait we had to go through and basically do a segmented annotation where we had to eliminate all this extra wax because the wax being present in empty cells in pollen and was really confusing the model when it says, I don't know what to do when there's wax present because you're telling me it's all of these things. So we were segmenting each individual cell so that we end up with a mask that is more exclusionary to this wax cappings that border the cells. And so when we add that approach in, we were actually seeing a huge increase in our percent accuracy for individual readings. So 20% increase for uncapped nectar, getting all of these readings into the high 90s. And we were adding in a few more features as well, like eggs, adult bees, and places where the foundation just simply was left undrawn by the bees. That way we can get a whole 100% estimate of what's going on in the frame. So we're still working on these areas. So like you can see, they're not quite as accurate as other features that we've worked on a little bit more. But ultimately our system, the goal is for us to put an image into the system and then have this image come back out with a full annotation where we can see, okay, here's a bee, here it shows up down here. We have 3 cells of capped brood here and one here. We're seeing those accurately show up. So a lot of confidence in the system when it comes to those measurements. And then the system basically will give you a percent coverage of each of those, which you can convert, to centimeters squared. The only case where we saw some decreases here in our model after retraining was in the drone brood. And what typically would happen is if it was misidentified, it was identified as capped brood. So still in the correct category. So I'm running out of time, but I just wanted to kind of highlight what our next steps are with the system. So really excited to be using this because again, there is a need and a call out for a system like this in regulatory studies. And our goal is to obviously maintain the highest quality standards that we can in the data that we are collecting. So we have our systems fully trained for yellow foundation, but we are working now to also add in scope black foundation frames as different regions have a different preference and sometimes these are used as well. And then the goal is to actually be using this set of systems in our regulatory studies for some of our first submissions that are going to be taking place in 2028 so that we can see how our regulators are going to respond to these systems. So with that, I would really like to thank you again for the opportunity to be here and to discuss this work that we are doing presently. Please scan the QR code if you want to gain your half hour of IPM CEU credit, and I would love to take some questions.
Thank you so much. And yes, please type in your questions into the Q&A portion down there. Just as I'm letting people do that, obviously this is really cool. It takes a lot more training. Yeah, I think sometimes we kind of think AI like, oh, it'll just fix everything. I was kind of amazed at the amount of training that it takes. I mean, do you think this tool could become available kind of outside the regulatory area, like for extension or beekeepers at a, outside of that area?
Yeah, it's a really great question. So right now it is proprietary as we're still developing and figuring out how to best release this information. We do have a goal of publishing this so that it can be available and so that regulators like EPA and EFSA can gain access to it and understand a little bit more what's going on behind the scenes. So hopefully that will happen in the near future. And then, once published, then it kind of is a public thing. So, but we're still making decisions on that. So I can't say definitively right now, but I would hope to see this become available because I think outside of regulatory studies where this is obviously going to be essential for high quality data, High quality data is essential for everyone, including researchers. And I also think that this creates a nice standardized system that, I myself did so many of these estimates in the field during grad school. And, I wanted to compare those to research that others were doing in different regions. And when we're not all using the same system, it makes that difficult. So if we can push for a system like this that's quite easy to use, a phone app that they can use it through so they can do it in the field, et cetera, that would be a dream.
Yeah, that's true. You think about, you train students to do these things and across the country, and there could be a great deal of variability. So I'll go to some of the questions. We have a question from Ryan Norton. Have you seen the AI and robotics work that BeeWise is doing today, using AI cameras and robotic frame manipulation that might be a way to enhance this study? They are tracking disease, brood production temps, burrow mite infestations. Might be a great collaboration for this work.
Yeah, absolutely. I think it's a great idea. We are aware of some the BeeWise work and a couple others who are doing some similar work. So we're not the only ones working in this space. I don't want to claim any credit in that way. And yeah, I think there's definitely some potential for future collaborations that can expand on not only colony population monitoring, but yeah, health metrics, disease metrics. So I hope to see that that is a path that we take in the future at some point.
Our next question, can the model reliably differentiate between capped brood versus capped honey?
Yes, very well. So our biggest issues right now are not necessarily in anything that's capped. The capped brood, the cappings on brood just look so different from capped honey that those are always coming out very reliably correct. Our biggest issues are actually in developing larvae at the very young instars compared to a cell that's just starting to be filled with nectar. Because in a lot of cases, it just looks like a little liquid in the bottom of the cell, and it can be difficult for the model to differentiate between those. So that's where more of our challenges lie. Anything where it's a wax capping versus that kind of grainy capped brood capping, the model performs super well in.
Again, please type in any questions into the Q&A. With your target of 2028 as your first regulatory submission, I hadn't thought about, regulatory agencies needing to accept methodology. Are there other instances where we're using AI tools, or is this kind of the first time for this?
Yeah, that's a great question. Like I said, the brood development tool, this is a tool that's actually already being used in regulatory studies, and not just by Corteva, by many industry representatives. And so, yeah, I think there's a precedent for it already. What we like to see is that we have a good system in place, and then that system is publicly available, because this is not something really that regulators can say, we require you to use this if it's not something that people can gain access to. So once you get to that stage with those types of systems, which the brood development system is at, absolutely they are being used in regulatory studies. So we expect that since there's a precedent in place for that, that this should be a pretty smooth process. But we'll continue to obviously engage with and have conversations and educate both ways with regulators and industry.
Oh, and we have a great question. Can you give an example of like what is a regulatory? I actually have no idea what's a regulatory study.
Yeah, it's a super great question. I feel like regulatory work is really complicated to understand for the general public in a lot of cases because we don't have easy access to understand what those requirements are. So in the specific instances of the studies we do, we're doing lower tier tests at first pass. So this would be a laboratory study with a specific set of organisms that represent a surrogate for specific functional groups. And so pollinators is obviously one of those functional groups. And so we have specific honeybee tests that are required to be run, bumblebee tests, in some cases solitary bee tests. And that varies depending on if you're registering an insecticide or an herbicide, fungicide, what those requirements are, but they are strictly laid out by your regulatory agency. So EPA for North America is going to have a specific set of requirements where if you are registering X product, you're required to run a study that shows that your product at whatever level you're planning to introduce that into the environment is not going to cause harm. So that would be a hazard test. And so basically getting an endpoint for an LD50. So what proportion, at what concentration is 50% of that population going to experience a mortality event? And then if we see some effects at these lower tier tests, it can trigger higher tier tests to say, okay, instead of giving them this range of doses that might not be totally representative of what you'd see in the environment, let's take it to the environment and see how it works in the landscape. So that's when we trigger these higher tier tests. And in certain instances, they're absolutely required. Like I said, in some cases, they're conditional. So those documents, that EPA guidance on like, here's your sort of checklist of what you have to do, are publicly available. Anyone can look up what that list is. And those studies are done in a super high quality and rigorous way. So following a complete good laboratory practice where everything is tracked, everything is measured out a very specific way. All of the analytical is confirmed, so we know we're giving them the doses we say. And that's where I think these AI systems, that's where the most confusion comes in, is that we've had a lot of questions from people about, well, how do you know the system is working such that if you put an image in, you know you're getting the right answer out? And so in the similar way that we would say, hey, we gave them X dose, and so we would have to run that through some kind of GCLMS to say, hey, we analytically confirmed that that dose is what we said. We do the same for these images where we would calibrate them with a set of images that come from the study, run them through, create a calibration curve. That way, if we have to do any adjustments on the numbers that the model gives out, we know that they're true to the system because in certain instances, the lighting may vary or the settings of the camera, depending on who's taking them, might not be exactly the same, and those images may vary. And we want that system to work reliably in all of these scenarios.
Yeah, that kind of leads a bit into the next question, which are there requirements for the quality of the images used with this process?
Yes, so we have developed fully SOPs around how the system is to be used, what the requirements are. And this took a lot of work where outside of training images, so much more went on in the background where we were out in the field with beekeepers, with cameras, testing out, okay, at what angle do we need to do it? How far from the frame do you need to be? How much of the frame do we get in there? What level, quality level does it need to be as far as how much light comes in on the camera and what the f-stop is? So I learned so much about photography through this process. It was really fun. But yeah, there are definitely some strict set of rules of minimums that must be met in order for an image to be quality enough. And that's similar. We took a little bit of information from that already developed brood development assay I was talking about because they have some similar kind of requirements that allowed us to kind of get an understanding of what we would need as a baseline. And then obviously this is something that we would go through with every study and say, okay, are we going to meet the requirements ahead of time? Do a couple of test photos. But yeah, you do want to maintain a specific quality in order to get high quality output. Like I said, the quality input dictates what you're going to get back out of it.
Next question, is it possible to measure colony strength at the hive entrance without taking pictures of the combs?
I think that probably there's a world in which this is possible. So there's a lot of work that's already being done in certain areas where bees are barcoded, and so you can track individual bees. Um, Certainly, I think there is potential for that. The reason that that's not necessarily something we sought for this is just the level of work that would have to go into preparing a colony for that in advance, because we wouldn't be able to differentiate, if a bee is coming in and out, which bee that is. And there's also always going to be a proportion of the colony that just isn't entering or exiting the hive, right? So we have our foragers coming in and out, and maybe some undertakers that are coming out to remove dead bees, et cetera, and those that are guarding the entrance. But there's a whole cohort of bees that are developing still, nurse bees that are just not leaving the colony yet. And we wouldn't be able to capture all of those with a system like that. And so what we're finding with this system, where we can just pull a frame out in the same way the beekeeper would pull that frame out and just take a quick estimate. Hey, this is 50% coverage. Our goal is to be able to take that picture just as quickly and then put the frame back so that it's minimal impacts on the hive, basically resulting in the same level of disruption that has already been historically happening. And we have actually found that compared to a beekeeper going through a colony, we side by side did photos, and it takes around the same amount of time. It's approximately 10 to 12 minutes per colony to get all of the photos captured then we can bring those back to the lab and do all of the post-processing later. But then you also have an archived copy image, right? Rather than just taking that estimate in the field and hoping that that's right.
Very good. There's any other questions? I'll let anyone type in questions as thank you, Ashley, so much. That was so interesting, and I'm kind of excited to see. I'm as a person who's done lab work and counted nematodes and realized wait, I'm wildly inaccurate probably through the day, let alone comparing to other people. I think these sort of tools are going to be really beneficial to research where you're trying to compare things,
Yes,
across multiple
absolutely.
things.
And
So
we're
very
hopeful to be able to use systems like this. I think this is a really great way to advance this type of technology. So we're, working in other areas, not just pollinators. I know we have some plant bioassays, some collembola bioassays, et cetera, where we're trying to adopt a similar process of image analysis detection so that we can speed these up, help reduce costs, but most importantly, increase that accuracy and precision that you're getting in those measurements and not require a certain level of training by any one individual employee that does those.
Very good. Again, anyone feel free to type in any questions. I just want to let everyone know that our next Pest in Progress webinar will be August 19th at 12 p.m. Central, 1 p.m. Eastern. Dr. Ashley Leach and Dr. Natalie Constancio from The Ohio State University are going to talk about from detection to action, environmental DNA, 3D printing, and grower outreach for spotted lanternfly. IPM. And so that will be in 1 month. And be sure to watch our newsletter and you will get information on how to register for that. Thank you so much, Ashley, and have a great rest of your summer, everyone.
Yeah, thank you so much for having me. It was wonderful.
Thank you.